Okay, so thank you everybody for your interest in our webinar. My name is Louis Bond. I'm the chairman of the ReSViNET Foundation. And today we have a very special webinar that I will introduce in a second. Everybody, good morning, good afternoon, good evening, wherever you are. I'm very happy that you're here with us. First, I would like to share our vision to connect global expertise and leadership in order to decrease the global burden of RSV. Our mission is to support performance of high-quality research and to improve knowledge of RSV epidemiology and to develop safe and effective therapeutic and preventive interventions. Our goals are to advocate and create awareness for RSV disease, to act as a focal point for effective partnership with stakeholders, and to combine knowledge and capacity required to enhance the development of novel RSV therapeutics. Next slide. We have had a series of RSV roadshow webinars together with the WHO and PATH. In the web events program, you can see that we have a number of webinars planned for January, February, and March that you are very welcome to participate. Please register, and these are well evaluated and highly interesting webinars for policymakers, but also for other stakeholders in the field of RSV. Next slide. So today's webinar is about the discovery and the development of first in RSV drug therapeutic Ziresovir. Next slide. Before we start and give the floor to the presenter, I would like you to use the Q&A button on your screen to submit questions. Please do so during the presentation so that at the end of the presentation we can use these questions to interact with Jim. So the Q&A button is there for that purpose. Next slide. So today's meeting will be presented by Dr. Jim Wu, who is the founder and chairman and CEO of ARC Biopharmaceutical. The company is designed to the development of innovative drugs for respiratory diseases and pediatric diseases, and they have a rich pipeline of for respiratory viral infection, pulmonary fibrosis, and other pediatric diseases. And I would invite you to take a look at their website. Next slide. I would like you to enjoy the webinar, and I will now give the floor to Dr. Jim Wu. Go ahead, please.
Well, thank you very much, Louis, for kind invitation and introduction. My name is Jim Wu. I'm a biochemist by training, actually working in industry for close to 30 years. So today I will tell you our story about the journey of discovery and development of the Ziresovir. I think everybody in the audience probably have expertise in the respiratory disease area, so I will not say too much, but I think respiratory diseases, especially respiratory viruses, still is a sustained threat to human health, especially after COVID outbreaks. I think even in the news media, you will see people talking about the so-called triple demics of COVID, flu, and RSV. So I want to mention for among three viruses, COVID and flu all have its very effective therapeutic drugs. However, RSV still does not have an effective drug. So this gives us a task to develop a drug to treat the RSV infection. So if we look at the history, actually the long history of our pharmaceutical industry and the biotech for the anti-RSV drug discovery, it was a long journey. We know ReSViNET was approved back in 1986 for treatment of RSV infection. However, the drug was not very effective. It also have some genotoxicities. So it's not even generally recommended by all the guideline right now. The first antibody for prevention was invented in 1996, I think, by the George Sieber. But now, because we have a better monoclonal antibodies, so ReSViNET is already in the history. But so the next really the major milestone in the discovery development of anti-RSV drug was really the Synagis, approved in 1998. It was quite effective. However, it required multiple injection during the RSV season, and also is highly very expensive. Pre 2023, there are many efforts have been failed during the drug development. So that promote us to put efforts for the anti-RSV drug discovery and development. So our efforts actually was initiated back when I was at Roche R&D center in China. So at that time, we recognized a huge medical need for RSV infection, and we feel we should do something. So we initiated the discovery efforts by set up a high throughput screening, use a HEP2 cell and RSV infection in a high throughput format. Basically, we screen the whole Roche, I think 1 million compound library, looking for the compound that can stop RSV infection to the HEP2 cell. That's how we identify a very interesting hit, showing on the right top of the figure. However, that molecule still doesn't look like a drug, because it has this the primary amine hydroxyl group, which is a little active, and also EC50. It is still not ideal with 0.22 microgram, and the safety index is low. So our chemists, I think close to 20 chemists, worked 2 years to optimize molecule, come up with a preclinical candidate. At that time, we call AK0529, with a 3 nanomolar potency and very high safety index. I think what we did not report too much in the literature is a very balanced PK tox profile, because sometimes, when we're talking about the drug potency, physical chemical property probably even more important as a drug which can go all the way to the end. So with this molecule on hand, first we try to determine the mechanism of action, because this is a compound get from the high throughput screening. We don't know the molecular target of the molecule. So we did the drug resistance screening and identified the mutation in the F protein, so that we know the drug actually bound directly to the RSV F protein. The crystal structure already been solved. It's specifically bound to the pre-fusion conformation of F protein. So what the molecule does, first, it can inhibit the virus entry to the epithelial lung cell. Secondly, it can stop the F protein-induced viral transmission through the cell-cell fusion, or we call syncytial formations. So mechanism of action of the drug, discovery of the molecule has been published in Journal of Medicinal Chemistry, I think 5 years ago. So if anybody interested, you can take a look at the paper. So since the molecule was discovered, we did extensive preclinical studies and also the clinical study now. The clinical development of the Ziresovir was initiated back in 2014 when we started the first in-human study in Australia in healthy volunteer basically is a typical so-called single ascending dose study and multiple ascending dose study to look at safety, tolerability, PK, and the food effect on the molecule. And since there, we conducted other phase 1 study, including a similar phase 1 study in China, a mass balance study is a C14 labeled molecule in UK. We also did the drug-drug interaction studies and some other safety clinical pharmacology study. So here just list the major study we conducted so far. So till today, more than 400 subjects has already received at least one dose of Ziresovir. When we develop a drug to treat RSV, I think the major question we are facing is what primary endpoint we can use for the clinical study. We know like J&J, Gilead, even Enanta use their own score system. We are a small biotech company. we cannot create our own score system. So we look at all the available score system. We feel we like the WHAM bronchiolitis sign symptom score. The reason we like it is first we look at the FDA guideline. We feel the major sign and symptom caused from the low respiratory tract infection are covered by WHAM bronchiolitis score. And also it's very simple. It's only have a 4 subcategory, and it's quite objective. If we look at it, it's a respiratory rate, respiratory muscle retraction, wheeze, and general conditions. So here I also listed the RSV ranking score, I think used by Dr. John DiVincenzo in the viral challenge study. But I would say, if you look at that score, it's a very comprehensive, but it's mainly a patient-reported outcome, and also it's including some upper respiratory tract infection symptoms, which is highly valuable in a clinical trial. So we feel in order to be successful, we need to use a so-called foolproof sign symptom score so that all the PI from different country, different hospital can easily assess the severity of the disease objectively. The phase 2 study, proof of concept study, was carried out in RSV infected hospitalized infants. So this study was conducted in mainly we enrolled the patients from 5 country regions, including Australia, Taiwan, Israel, Malaysia, and Turkey. We enrolled 72 infants patients. So here just shows some basic information of the trial. Basically, we randomized the patient 2-to-1 ratio from active drug to placebo. In single dose study, we tried a different dose level, look at the PK, PD correlation. In part 2, we conducted a multiple dose study, dose drug BID for 5 days, and also tried a different dose level. The main result has been published in influenza and other respiratory viruses last year. But the real result showing on these 2 figures, first is antiviral drugs. So we look at the viral load reduction in the 2 milligram per kilo group versus placebo. We do see 98%, so close to 2 log reduction, additional reduction compared to placebo patients. So that demonstrates the drug actually works, can reduce viral load in the patients. On the right figure, we measure the WHAM bronchiolitis sign symptom score, also at the 2 milligram per kilo group. Compared to placebo, it's reduced the sign symptom score by 4 points versus placebo group. By the end of day 4, it only reduced 2-fold. So we feel the drug has the potential to show the clinical benefits in the patients. The phase 3 clinical study was conducted in China only because we conducted it during the COVID time, and we are certainly we will not be able to do a global study. So this study inclusion exclusion criteria is very similar to the phase 2 study. We enrolled the patient from 1 to 24 months old, RSV positive with IC symptom less than 7 days. RSV diagnosis confirmation less than 1.5 days. And I think another important criteria is sign symptom score has to be above 5 in the scale of 12 of the WHAM benchlider sign symptom score. So these are the, I would say, moderate severe infected infants. So these patients are randomized 2-to-1 ratio, and part 1 study, because it's first time we dose a patient in China, so we did a safety PK study to 54 infants patients. Part 2, for the efficacy assessment, we enrolled 248 patients. We dose a patient BID for 5 days. It's a weight band dosing. We evaluate the sign symptom score and also collect virus specimen on a daily basis. Once finished 5 days, we have 9 days follow-up. So the primary endpoint is a WHAM branchiolitis sign symptom score change at the end of 48 hours after first dosing of the drug. The reason we set up 48 hours certainly based on the phase 2 result, and also we feel for a direct antiviral drug, if you don't see the efficacy within 2 days, generally physician will switch to another supportive care. The key secondary endpoint is viral load reduction at the end of day 4. So this is the EFLO study baseline and demographic summary. I think these are the typical RSV infected infants. 2/3 of patients, I think, under 6 months old, because they have more severe disease. They are more willing to participate in our study. Most are the male. I guess the boy are more susceptible to RSV infection, have more severe disease somehow. They have a very normal weight. RSV onset day on average was 4 days. Actually, I just recently saw the Inentus data. I think it's very similar. It's RSV onset day is day 4. Baseline benchlight score was around 7. Baseline viral load was 7 log copy per mil. So it's very similar to the number in the literature. We included patient infected with subtype A and B, about half and half. We did detect a third of patients have other respiratory viruses if you do a PCR detection. But we don't think other viruses actually play a major role in this respiratory infection. We also including about 10% of immature baby and about 4% of ICU patients. So the phase 3 study result has been published in New England Journal of Medicine, I think 2 months ago. We are very grateful they accept our paper, and New England Journal of Medicine also published an editorial by Dr. Walsh to hail the positive result of phase 3 study. So in the following slide, I will show you the main result we got from this phase 3 study. So if we look at the primary endpoint at 40 hours after dosing, so we can see the active dosed group have reduced sign symptom score by 3.4 point. Placebo was a 2.7 point. P-value, I think it was very good, 0.002. If you look at every data point, we have a standard deviation, actually quite small, indicate a high quality of the data. So compared to placebo, active dose group reduce the sign symptom score by more than 30%. We certainly examined the subpopulation. Here just shows some of the data. So we look at the infants under 6-month-old. So at 48 hours, actually reduced 1.3 points, 1.2 points. So compared to placebo, it reduced the sign symptom score by more than 55%, which so indicating the little one, the more severe one, will benefit more from the treatment. We also look at the four sub score of a one benchmark score, including respiratory muscle retraction, respiratory rate, wheeze, and general condition. And you can see compared to the placebo, all the category got improved with actually a statistically significant effect. And the younger one improved even more in 4 categories. So that means the drug effect actually is on all aspects of the disease. We also examined a secondary endpoint is a time event to the 50% sign symptom score reduction. So active dose group did improve the symptom 1 day early than placebo. We also look at the respiratory rate reduced normal. It's also have a 1 day improvement. Time to the subcategory general condition score reduced to 0. I think we also have a 1 day improvement, although the p-value is a little bit high. But you can see the trend. There's improvement compared to placebo. We also look at the disease remission. We define disease remission by reduce the sign symptom score to 1 to 0 without a non-invasive positive pressure ventilation or mechanical ventilation or assisted oxygen therapy. You can see there's also a 1 day's improvement, although the p-value is a little bit high. There's only 12 subject ICU patients was enrolled in our study. So I think certainly it's not the power to draw any conclusion, but nevertheless, we look at the stay of ICU. We found the active dose group have a median stay of 3 days versus placebo group of 8 days. So this suggests treatment of the RSV infected ICU patients could dramatically reduce the ICU stay. So previously, these are all the clinical endpoint. We also examined the viral load change, and viral load is in the unit of a log. So 1 unit equal to 10-fold difference. If you look at it, I think it's quite significant. After 2 days, you can steadily show the antiviral effect, because this viral load is determined by the PCR method. So PCR method, it does take time for the virus cleared from system before show the drop of the viral load, because it can detect the dead virus too. But nevertheless, you can see from the second day, we see a dramatically reduction of viral load compared to placebo. At the preset secondary endpoint time at 96 hours, we reduced 0.6 log, which equal to 77% reduction in viral load. We also look at the percentage of subjects with viral load below the low limit of quantitation, and the blue bar showing the active dose group play bar showing the placebo, you can see the treated group have a much high chance to reduce viral load below the low limit of quantitation. Since this is an antiviral drug, certainly we have to look at the emergence of drug resistance. So basically, we did detect drug resistance, the mutations around the drug's binding site. But these all appeared, I remember, is post day 4. They start to appear. Actually, antiviral effect, even the clinical benefit appeared much earlier than day 4. And overall, about 8% of the subjects have the drug-resistant mutation. So that confirms the mechanism of action of the drug target on the RSV F protein. And also, I think the good thing is RSV is not highly mutated virus like influenza. It is highly mutated, but not like influenza, which every year come up with a new strain. So I think, and also we look at these mutations. I don't think we found in gene banks. So this is a good thing, good news. Safety certainly is very important, especially the drug is intended to treat infants. If we look at the drug-related AE, they are very low. It's very comparable to placebo. Even we look at the specific AE, diarrhea, liver enzyme elevation, rash, platelet count elevation, and so on. They are, I think, probably there's some AE associated with drug, but overall is very mild. We did not see any drug-related SAE, certainly no treatment discontinuation, and all other severe events. So the drug actually is quite safe. In this study, we dosed 200 infants. I think quite a few are as young as 1 month old. In this phase 3 study, we actually also did a 2 years follow-up because our leading PI feel, well, for antiviral drug, you always show a short-term effect. So what's a long-term benefit? So we followed the most of patients, look at the recurrence of wheeze. Very interestingly, in 2/3 of patients who are under 6 months old, we see the annual rate of recurrence of wheeze reduced significantly in the active dose group compared to placebo. I think reduced by 3-fold or 2.6-fold. Try to remember the number correct. So that means for this quickly developed infants, I think the virus infection do have some damage to the respiratory tract. Early treatment of drug have a long-term benefit to reduce recurrence of wheeze from our study. We also look at the all this antiviral drug to treat the acute viral infection. We find that they are kind of similar. If we compare to the Paxlovid or compared to the Tamiflu, the viral load effect by antiviral drug actually is quite similar. The clinical benefits is also kind of similar because acute viral infection only lasts for a week or so. So whatever you can show the short-term benefit probably is 1 day or 2 days or reduce sign symptom score by 50%. But nevertheless, I think the commercial potential is huge. This is why it's motivated us to work hard in this area. So this is the last slide of my talk. Basically, the drug already completed first phase 3 study. We are currently doing a phase 2 study in China. We are supported by government's plan for so-called category of a major new drug invention. We also granted breakthrough designation, actually first ever pediatric drug received breakthrough designation from China CDE. We also granted a priority review by the China CDE as well. So hopefully the drug can get approved to use to treat the very needed infant patients. We're also talking to FDA right now, try to finalize a global clinical development plan. So that's all for today's talk. Thank you for your attention. Well, certainly we need to acknowledge the patients, their parents for participating of the trial and support, and all the investors, clinical staff, CRO, collaborator, our advisors, and our team for contribution and efforts. Thank you.
Thank you so much, Jim. That was a fantastic overview from where it started till where we are now. So, and thank you so much for staying in time. A lot of folks have shown up, including a lot of people that I know from the field that are knowledgeable and know exactly how difficult it is to get to an antiviral drug developed until this stage. So congratulations to everybody on the team and all your colleagues. So while people use the question and so the Q&A button, I would like to start first with a question from Michael Rodin, who actually thanks you for the talk and wants to know the phenotypical consequences of the mutations that you showed. You showed 5 patients under treatment that developed mutations. And have you done any efforts to understand what the effect has been on of these mutations?
Right. So in our clinical study, we certainly look at the viral load. We did not see any rebound of the viral load in this patients with a drug-resistant mutation, because we know eventually it's our immune system clears the virus. The drug just help to stop the replication. So it's a dynamic equilibrium. So once you stop replication, the immune system will clear the virus. So whether it's a mutant or not, it will eventually to be cleared. We did not see any rebound.
And did you use reverse genetics to recapitulate the virus and study it in vitro, such as fitness and susceptibility to drug, etc.?
Yes, it's required by FDA. So we did it too, made the recombinant virus. These virus mutants actually have been reported in the literature as well. Certainly we did our experiments too. So I think the fitness is not much different from the wild type. It's a behavior like a normal virus.
And susceptibility to drug is gone, probably, right?
Yeah, it's gone. It's gone.
Yeah.
Yes.
So maybe the question that Michael is also suggesting is that if the viral fitness is normal, then if we start using Ziresovir more extensively, what is the risk that this virus is for some reason going to become the dominant strain and that we lose the drug as a treatment?
Right. So this is an excellent question. Certainly, we consider it every day. But I think if you look at influenza, after the introduction of Tamiflu, I think only one season we see the emerging of resistance, I think in Canada, but these are mainly to do the antigen shift of the virus. I think the acute virus somehow, they just don't spread around the drug-resistant mutant. So far, we haven't seen the dominant emergence of drug-resistant mutants, even after 20 years of use of the Tamiflu.
Thank you. Yes, I think Tamiflu is a good example that it does not necessarily have to lead to widespread resistance, but there are other antiviral drugs for influenza that were leading to resistance that really ended the utility of that drug. So you brought up influenza. So what we know, while people have questions, feel free, folks. What we learned from influenza is that we have to treat early if we want to be efficacious. You talked a little bit about intended use and young babies, maybe more efficacy in the young babies. But do you have any idea in a few years, if this drug is really entering the market, what is the best use of the drug? Would that be in the early phase, the mild disease, or in the phase that children are at the hospital or even at the intensive care unit, as you have studied now? What is the best use of drug?
Right. So this actually is a great question, because we know for influenza, you have to treat it early, within 48 hours. For RSV, we heard some colleagues from other companies showing you have to treat it early in order to demonstrate some benefit. But in our case, actually, for our phase 2 study, it was a real life study. So we did not set up a disease onset time. Based on the belief, actually, is John DeVenture's beautiful work showing the symptom and the viral load correlate very well. So if infants have a high viral load, if you reduce it, the symptom score will reduced. So I think it will provide a benefit. For influenza, because these are the cytokine storm, so even you suppress the viral load, they're going to still have this inflammation effect. So I think certainly based on our data, you can even treat the patients with a much longer infection. As far as viral load is high, you suppress it, you will demonstrate the effect.
So as a follow-up question, did you treat children at the same time of infection? Probably the duration of symptoms before treatment was heterogeneous. And did you treat children 5 days after onset of symptoms, for example? And did you see differences in effects depending on timing of treatment?
Right. So in our phase 3 study, we set up a disease onset time under 7 days. So we did do the subpopulation analysis as I think a 4 day or 3 days cutoff. We actually did not see difference. Even the later treated patients show the benefits.
So what does this suggest to you? Does it suggest to you that the cytotoxicity or the virulence of the virus is the most important mechanism of disease severity? Because apparently, if you just kill the virus, the disease ends. Most people in the influenza field, like you just explained, think that the cytokine storm or the immune pathology at a certain point is the dominant mechanism of disease. Are we learning from treatment, in your case, that basically virus-induced direct viral damage is the main mechanism of disease?
Certainly, I think it is the most impact. Once you're infected, certainly, induce the inflammation, cytokines expression. But I think the cause of everything is really the RSV infection. We know some viral proteins actually can induce pro-inflammatory effect, for example. So if you reduce the viral load, certainly it should have the benefit.
Yeah. And so this is like asking for a vision, Jim. Do you have any vision on combination treatment in future? I think the risk of resistance will always be there. Whatever happens, I think there is a risk, regardless of the size of the risk. Do you have in your mind that combination therapy could be not only be more efficacious, but also prevent the development of resistance? Any thoughts?
Yeah, certainly, we work in the antiviral area, like the idea of cocktail with a different mechanism of action, as you suggested, can suppress the emergence of resistance. But I think the good thing is about acute viral infection, because it's a short period of time. So it may not have a huge impact versus a chronic viral infection. Certainly, you need a combination for HIV, HBV, HCV, you name it. But for acute viral infection, I certainly personally love the idea of the combination. That's why our company also work on different target. Now I think in Inenta have an N protein inhibitor. It'd be a great idea to combine them. But we have to persuade the FDA first, because the FDA probably want to approve a single therapy first before even consider the combination.
I understand, but taking that a little bit further, the immunocompromised hosts, children with cancer or with severe HIV, have increased risk of severe disease. Maybe not the dominant category, but I'm pretty sure that once it's on the market, people will start using it for patients with prolonged replication, and maybe very prolonged replication, because in the absence of an immune system, let's say bone marrow transplant patients, maybe for weeks and weeks, people will start using it. So that's like an ideal situation for the development of resistance. So did you actually, so there is a question from Sonia Mararaboni. Could you explain in your studies whether you've tried drug in immunosuppressed adults or children?
We haven't. We haven't. But certainly, I think this is a high-risk population is worthwhile to explore. Because if you look at the patient population can treat, infants, I think, is the ideal one because they don't have a comorbidities. They are very clean. So it's an easy to demonstrate effect. But I think, as you suggested, the immunocompromised one is really the patient needs the drug. Once they have RSV infection, it could be very dangerous. So we are doing some studies in China, try to look at the disease cause, what we can do. But I cautiously optimistic, because if you look at the previous Gilead's work or J&J work, it do shows a good sign to reduce lower respiratory tract infection in these patients.
And did you see any differences in efficacy or resistance element A versus B? Did you see more? Go ahead.
Right. We haven't have the data yet. Hopefully, one day we will have a data to show the public. Yeah, we don't have right now.
Yeah, very important question. Thank you, Michael, for your active involvement. So before we ask my colleagues at the ReSViNET Foundation to turn on the presentation on our behalf to share a little bit of our chatbot and information, I have one final question for you, Jim. What does it taste like?
Oh, no taste at all. Well, a little bit sweet, I would say. So I personally tasted the drug because we know it's going to those to infants. So there's no taste ever. Because our drug, I can tell you some secret. So this drug is coated on a sugar core. So there's a little bit of sweet taste. So I think perfect for infants patients.
Thank you. That is a wonderful closing statement of this webinar. Thank you so much for making time to inform so many people about what you're doing. And it was wonderful to see how your plans have come to fruition in your recent New England Journal of Medicine paper. So before we close this session, I would like to inform you about the RSV chatbot that we have generated for the general public. And we know that a lot of people are now becoming more and more interested in RSV. So if you go to the next slide, we have developed a chatbot that is continuously available that interacts in all languages. So please use your personal language of your favorite language to try it out. And at the bottom, you can see that it is expert verified content. So we are convinced that what you will find in this artificial intelligence based chatbot is actually valuable information that people can use to be informed about RSV. So the general public, all those billions of people that now need to know about prevention and treatment of RSV, they have access. So if you go to the next slide, I would like you to take your cell phone now, and I'll pause here, and you make your way to the chatbot using this QR code. And if you use this QR code, you can start interacting through WhatsApp with the chatbot. And we would love to hear your feedback, to hear whether or not you find this information useful, whether you feel that the information was correct, if that is what you're testing, and if you have any advice or suggestions to improve it. We're actively working on it, and we get really good feedback. Thousands of people are now starting to use it, so we are really curious about your thoughts. So please share it with your colleagues. We think this is it's going to help. It is developed completely independent developed by the foundation, independent from any other support, together with the patient advisory board. Next slide.
There is no next slide.
Right. So this ends our webinar, and I would like to thank you all. In particular, Jim, thank you so much. I will do the applause on behalf of everybody, and I wish you a great day. Thank you for joining, and see you soon during one of our webinars. Have a great day. Bye.
Thank you. Bye-bye. Thank you.